This video will go through subtopic 1.6, cell division. So here's a list of understandings, application and skills, and guidance points that we will cover in this video. So when we talk about cell division, we have to first talk about the cell cycle, which will ultimately lead into that process of cell division. So there are three functions of cell division. It's used for growth, for repair of tissues and organs, to maintain and replace dead or dying tissues. It's basically how cells within your body get replaced. So the cell cycle takes about 12 to 24 hours for animal cells, depending on the cell that is being replaced. So ultimately with cell division, it's about replicating the DNA and then dividing that DNA between the two cells now, or getting an equal share of DNA into that new cell that is being created. So in animal cells, the DNA is rather large and linear, so that DNA does need to be packaged into a more into a structure that allows it to be divided up easier and to ensure that the DNA is divided equally. So prior to cell division, a process of supercoiling of DNA will happen. So during mitosis, the two chromatids that make up each chromosome have to be separated and moved to opposite poles of the cell. So it's therefore essential to package those chromosomes into shorter structures and into more condensed structures. So condensation of the DNA occurs by coiling these DNA molecules to make up these chromosomes. And the chromosomes are going to be shorter and wider through that process of supercoiling. And during that process of coiling, you have these proteins called histones that associate with the DNA and help with the supercoiling. The image on the slide shows how this coiling happens. So you have just the basic structure of DNA. The DNA will coil around those histone proteins. And those histone proteins now coil around each other. And then ultimately you get more coiling, and eventually you form these structures of chromosomes. And it is the chromosomes that will be involved in the division of DNA during cell division. So in the cell cycle, there are three major phases to it. There is interphase, which can be divided into three smaller phases. So there's the G1 phase, S phase, and G2 phase. In the G1 phase, this is where the cell grows. It increases the amount of cytoplasm and organelles. Once it reaches a certain size, then DNA replication will occur. This is where an exact copy of the DNA is now made. And it ensures that new cells have equal amounts of the DNA. Once the DNA has been replicated, the cell can then enter into the G2 phase, which is additional growth and more preparing organelles for division, and more mitochondria get formed as well. Once the cell has grown some more and is then ready to enter into mitosis. So that process of mitosis is going to be that nuclear division or DNA division. So the DNA will get divided between those two cells. And mitosis will ensure that the same number of chromosomes are given to each cell. So a cell can't have too many chromosomes, and a cell shouldn't have too few chromosomes. So there's an exact number of chromosomes that are needed, and mitosis ensures that each cell gets the exact number of chromosomes that are needed, or should ensure that they get the exact number that are needed. In the final stage of the cell cycle is cytokinesis. This is where cell division occurs, and where you have division of the cytoplasm. So in plant cells, this is where the cell wall forms. And in animal cells, you get a cleavage furrow that forms that separates the two cell membranes. So just kind of before we go into the stages of interphase, or mitosis, we'll first look at interphase. So in interphase, the DNA in the cell exists as a structure called chromatin. Chromatin is going to be an uncondensed version of the chromosome. So this is where the DNA is uncoiled and easily accessed. The nucleolus is going to be surrounded by a nuclear envelope, so we'll have the presence of a nuclear membrane. And during that G2 phase, you're going to have centrioles that are duplicated. So when viewed under a microscope, typically what you see is this darker structure which would represent the nucleus. And it's not going to be very dark, it should be noticeable, but it appears darker just because of that DNA that is present, and that is unwound. So once a cell is ready to leave interphase and then enter into mitosis, you're going to have the parent cell that divides ultimately into two genetically identical daughter cells. So the whole process of mitosis is to create these two genetically identical daughter cells. So the results of mitosis is that you get two daughter cells that are identical to the mother cell or identical to each other. And this process of mitosis is going to occur in somatic cells. So somatic cells are basically any cell that forms the body of the organism, excluding reproductive cells. So reproductive cells would be considered gamete cells. So this would be, for example, sperm in a male or eggs or ovum in a female. So if you're not a sperm or an egg, then you're a cell that makes up the body, and you are considered a somatic cell. So liver cells, heart cells, muscle cells are all considered somatic cells. Mitosis ensures that all these body cells, all these somatic cells, will have a complete number and form of chromosomes. So in humans, in all human cells, the result from mitosis will always be what's referred to as a diploid number of 46. Diploid meaning two sets, and the 46 means that there are 46 chromosomes in total within that human DNA. So all cells that all human cells that undergo mitosis will end up with 46 chromosomes in the end. So this picture just gives an overview of the different steps in mitosis. Basically, you're starting with interphase, where the DNA is unwound and uncondensed. And as it enters into mitosis, the DNA now condenses into those chromosomes. Those chromosomes line up in the middle of the cell, and then those chromosomes get split. Once those chromosomes have moved to opposite poles of the cell, then you get reformation of that nuclear membrane, and you get formation of that cleavage furrow. So you get separation of those two cells. So looking in detail at the specifics of each step of mitosis. So the first part, or the first step in mitosis, is going to be prophase. And this is where the chromatin supercoils into those chromosome structures, so that DNA condenses into those chromosome structures. Two chromosome arms are generally going to be referred to as sister chromatids. And so when we say chromosome arm, if we have a look at our diagram that I just pointed to, a chromosome arm would essentially be what I've outlined in blue. So one half of that chromosome would be considered a chromosome arm, and then the two arms together would be referred to as a sister chromatid. And those sister chromatids are joined at a point referred to as the centromere. So the centromere will connect those two chromatids together. In prophase, so once the chromatin is supercoiled into the chromosomes, the nuclear membrane will then break down, and the centrioles that have been duplicated now will move to opposite poles of the cell. So these centrioles will then move to opposite poles of the cell. So when looking at a cell, when looking at a microscopic image of a cell and trying to identify what stage of mitosis it's in, you can generally tell that it's in prophase, or things that suggest that it's in prophase is the nucleus is not as uniformly stained as it would be in interphase. You can get a sense that there's kind of this blotchiness to the staining, which suggests that the DNA is starting to condense. In the next phase, metaphase, there are these protein structures called microtubules that attach to the centromere of the chromosome and will guide the chromosome to the equatorial plate, i.e. the center of the cell. So basically in metaphase, you get the chromosomes that line up in the middle of the cell. You have these microtubules that will extend from the centriole and will attach to each chromatid. So you'll have two microtubules attaching to each chromosome, or to each sister chromatid. So in metaphase, in the microscopic images, basically what you would be looking for is these chromosomes, or these sister chromatids that are now essentially lined up in the middle of the cell, and they appear, when the DNA appears, condensed in nature, as opposed to other cells that are not undergoing mitosis, where the DNA is more spread out. Once those chromosomes have been lined up, the next stage of mitosis would be anaphase. So this is where the sister chromatids are separated and then moved to opposite poles by the microtubules. So the microtubules that are attached to each sister chromatid, or attached to each chromatid, will now start to contract. And this will separate the two sister chromatids. Once those sister chromatids have now been separated, they now get referred to as chromosomes. As those microtubules contract, each pole will now have a complete set of chromosomes. So microscopic images of a cell in mitosis at that anaphase stage will look like, we'll basically have two sets of chromosomes now moving to opposite ends of the pole. And it should, I guess, kind of look like fingers extending towards the center of the cell, but there should be distinct staining in two sets, or two areas of the cell. In the last phase, which is telophase, this is where the chromosomes have now reached opposite ends of the cell, and the chromosomes will now start to unwind and become chromatin again. the microtubules break down and disappear, and we get the nuclear membrane reforming. So identifying telophase, you're basically getting what looks like two cells that are basically joined together, but they're two cells that are essentially separated. So you can start to see that separation of those two cell membranes. During that process of mitosis, you should be able to calculate that mitotic index. So that mitotic index is going to be the ratio of the number of cells in mitosis in a tissue and the total number of observed cells. So basically, it's going to be a ratio, or that percentage of cells that are undergoing mitosis in a sample of cells. So when you're viewing a tissue sample under a microscope, and you can observe all these different cells, you're basically counting the number of cells that are in mitosis as a ratio to the total number of cells counted in that field of view. And that mitotic index then is multiplied by 100. So obviously, the higher the mitotic index, the more cells that are divided. The lower the mitotic index, we have a lower number of cells dividing. And the last part of the cell cycle is cytokinesis. This is where you get division of the cytoplasm, and this will result in two genetically identical daughter cells. In animal cells, you have contractile proteins, actin and myosin, that pinch along the cell equator of the cell, and this will form a cleavage furrow. And ultimately, this will lead to separation of those cell membranes, and you get the two distinct cells that are formed. In plant cells, the process is slightly different. So you get vesicles that move to the equator, where they fuse to form these tubular structures. More vesicles fuse, and these tubular structures eventually merge to form two layers of membrane across the whole equator, which then develops into the membranes of the new daughter cells. A molecule called pectin is brought in vesicles and deposited by exocytosis between the two membranes, and cellulose is deposited by exocytosis to ultimately form that cell wall. Basically, the main difference between cytokinesis in a plant cell and an animal cell is that in a plant cell, instead of a cleavage furrow, you get the formation of a cell plate, which will ultimately be used to divide those two cells. So the following diagram just shows a summary of the cell cycle. So you have a cell that is in G1, where it grows, the centrioles replicate. From the G1 stage, it will then go into the S phase, which is where DNA gets replicated. After the DNA has been replicated, that cell will grow some more as it prepares for mitosis. Once it is finished growing, it will then enter into mitosis, undergoing those different phases in which the DNA condenses to form those chromosomes, moves to the equatorial plate. Those sister chromatids then get separated into chromosomes. During anaphase and in telophase, the chromosomes have finished moving to those opposite poles, nuclear membrane reforms, and then finally cytokinesis, where you get division of that cytoplasm. And then each cell is ready to enter into that G1 phase again to start the whole process over. So when looking at the cell cycle, it is a very complex process outside of the division of DNA. So there are molecules that will control the movement of the cell through the different stages of the cell cycle. These molecules are generally referred to as cyclins, and they do have control, or they help control the cell cycle. So cyclins are a bit of a complex molecule in terms of how they interact with each other. But basically, cyclins are going to bind to enzymes called cyclin-dependent kinases. Anything that is a kinase is going to attach a phosphate group to another protein in a cell. So those cyclins will bind to the cyclin-dependent kinases. Those kinases now become activated, and they will take ATP, remove a phosphate, and attach that phosphate to another protein. The phosphate that has been attached to that protein now triggers the protein to become activated and will carry out tasks specific to one of those phases of the cell cycle. So in order for a cell to progress to the next stage of the cell cycle, these cyclins have to reach a threshold concentration, meaning there needs to be a certain amount of these cyclins present within the cell. So the picture here demonstrates how these cyclins are working. So the cyclin itself will bind to another protein, and the CDK is going to be the cyclin-dependent kinase. When that protein becomes activated, it is going to take that ATP molecule and remove one of the phosphates. Remember, it's adenosine triphosphate. So it'll cleave one of those phosphate groups and attach that phosphate onto another target protein, or onto another protein. That phosphate being attached, becoming attached to that protein, will now activate this protein, or essentially turn on this protein to allow it to perform a specific function within the cell. So you need a certain concentration of these cyclins within the cell in order for the cell to move to the next stage, because you need a certain number of these proteins to be activated. So this diagram just kind of indicates, how cyclin might work in the cell cycle, and how it can act as a checkpoint, allowing cells to progress to each of the different parts. So cyclins do increase and decrease over the process of the cell cycle. Certain cyclins will trigger a cell to move into that next phase. So, logic might dictate that everything starts with cyclin A, but everything will essentially start with cyclin D, or will start with cyclin D. So cyclin D, as those concentrations of cyclin D start to increase, it will tell a cell to move into the G1 stage, and from the G1 stage into the S phase. As cyclin D increases, there is another cyclin that starts to increase, cyclin E, and cyclin E will help prep the cell for replication in the S phase. So cyclin E will allow the cell to continue moving into the S phase. As the cell progresses through the S phase, you have this other cyclin now that's building up in concentration, cyclin A. And cyclin A will activate DNA replication in the nucleus in S phase, and help push that cell into the G2 phase. And then as the cell continues to move through the G2 phase, you'll have cyclin B starting to reach a peak concentration. And cyclin B is going to promote the assembly of mitotic spindle, and will perform other tasks in the cytoplasm to prepare for mitosis. So as cyclin B reaches its threshold, it'll help push that cell into mitosis. So what I would say, the takeaway from this is that you need to know that there are cyclins. You need to know what those cyclins do. You should know what these individual cyclins do, but you do not need to know when each cyclin peaks or when that peak occurs. So don't bother with trying to memorize this diagram. So ultimately, the cell moving through that cell cycle is going to be a highly regulated event. That is the purpose for all these cyclins, carefully moving that cell through the cell cycle. So occasionally and sometimes the cell cycle can become unregulated and a cell can just start dividing uncontrollably. And this is when we can get a situation of cancer arising. So when you have cells that divide uncontrollably, this can lead to tumor formation. So tumors are abnormal groups of cells, and they can ultimately be categorized into two types of tumors. So there's benign tumors and malignant tumors. Benign tumors basically have cells that stick to each other, but the key part here is that they do not invade nearby tissues or move to other parts. Benign tumors grow to a certain size and then stop. They don't interfere with function of any organs and don't spread to other parts of the body. Whereas malignant tumors now, these are tumors that are going to invade surrounding tissues, affect function of those organs, and can even become detached and move elsewhere in the body. And when they move elsewhere in the body, they can develop into those secondary tumors. So what can cause that cell cycle to go awry? So this can be caused by compounds referred to as carcinogens and mutagens. So carcinogens are typically cancer-causing chemicals. Things that could be considered carcinogens would include like viruses, X-rays, UV radiations, some chemicals. Mutagens are agents that cause gene mutations and mutations that can ultimately cause cancer. So they can cause mutations in genes that would prevent the cell from dividing uncontrollably. And when you get mutations in those types of genes, it can lead to cancer potentially. So there are a type of gene referred to as an oncogene. Oncogenes are basically genes that when mutations occur to them will lead to the development of cancerous cells. So the thing to keep in mind is that when mutations occur to the DNA, it's basically they're going to be random and just happen by chance for the most part. So most genes do not cause cancer when they mutate. So if a mutation occurs to a gene, it's not necessarily going to result in cancer being the end result. So most genes fall under that category. There are a few genes, however, that can become cancer-causing after mutating. So if they mutate, they can lead to the development of cancer, and these are known as the oncogenes. So oncogenes are typically going to be involved in control of the cell cycle and cell division. So ultimately, you can get the formation of a primary tumor that is malignant, and at some point, that primary tumor can break away, or a portion of that tumor can break away and enter into the blood vessel where it can spread. So generally speaking, there has to be several mutations that occur in the same cell in order for it to become a tumor. Rarely is it just one mutation. There has to be several mutations that have to happen in a cell in order for it to become cancerous. This is why cancer typically shows up in older people. It can take a lifetime to acquire all of these mutations necessary for a cell to become cancerous. When a tumor cell has been formed, it will divide repeatedly and uncontrollably. So this group of cells is referred to as a primary tumor. So in the diagram, you have like that primary tumor that's growing. It can invade those surrounding tissues and invade that bloodstream. And you can have cells that break away from that primary tumor that then travel through the bloodstream and can end up in a different site. This is referred to as a secondary tumor, and that process of movement of those cancer cells is referred to as metastasis. So this is the movement of cells from a primary tumor to set up secondary tumors in other parts of the body. So typical locations where you get these metastatic tumors include like the brain, for example, the lungs, and even within your bones. So those are common locations for metastatic tumors to end up. So there is a strong correlation that exists between smoking and cancer. Cigarettes do contain a lot of chemicals in them that can cause a lot of damage to the DNA of cells, leading to mutations in the DNA over time. So there is that positive correlation between cigarette smoking and the death rates due to cancer. So in this case, the causal links are well established, so it's not like we don't know that cigarette smoking causes cancer. It's not a maybe it does, we know it does. So cigarette smoke can contain many different chemicals, and 20 of these have been shown to cause tumors in the lung. 40 chemicals, and we're talking like 40 out of like 200 chemicals, which is quite a bit, in cigarette smoke are carcinogenic, meaning that they can lead to the development of tumors. The diagram of the cigarette shows like the different types of chemicals that you can find in a cigarette. So you can find things like arsenic, ammonia, and of course like there's nicotine in there. There's often benzene, all kinds of compounds in there, carcinogenic compounds that when taken into your lungs can cause damage to those lung cells. And then the graph at the bottom of the slide just shows the number of years smoking and then the relative risk of developing cancer. So takeaway from this is the longer you smoke, the higher your risk of developing lung cancer. And especially if you smoke 20 plus cigarettes a day for about 20 to 30 years, your chances of developing lung cancer are significant. So there are some database questions on page 54 and 59 of your textbook that you should work on. So that's the end of this video for 1.6.